But maybe I will entertain the people who are already here with this picture, this photo that I just shared with you, László. So this is a true story. It's a picture I took in one of the Cambridge colleges in the library. I was just walking through the library and I recognized this familiar spine of a book. I'll just show you the photo. Um, so this is, of course, László's famous network science book. And then I had a bit of a double take because then I noticed, wait, this isn't the usual science section. And it turned out that someone had placed it in the Bible section. Um, so I think that's one good way of introducing László as a speaker.
That's fun.
Okay. And then I guess to introduce László more properly, he is the Robert Gray Doge Professor of Network Science and a University Distinguished Professor of Physics at Northeastern University, where he also leads the Network Science Institute. And more importantly, I guess it's fair to say that László is also a founding father of network science as we know it, and that his work has fundamentally changed and is still fundamentally changing how we understand connectivity and also complexity in the world. And I know his work has inspired many of us over the years. I thought I'd share one more personal anecdote, which is that I can still really remember coming across an article in New Scientist when I was about 17 or 18 about László's work on scale-free networks. And it really stuck in my mind. I threw away all the other back issues of the journal over the years, but I always kept hold of that one because I thought, not because I knew it would be relevant to my career later, but just because I thought this one's really interesting and I think I should think some more about it and maybe reread it. And I actually still have the copy somewhere. Um, so this is just one story out of thousands, but I know László has shaped, well, inspired many people, partly because of the science and also partly because of the effort that he's putting into communicating his work to a broad audience. So that includes writing a series of popular books, such as Linked, The New Science of Networks, The Formula, The Universal Laws of Success, and several others. And László also stands out as I think, an unusually bold communicator in that he conveys both the science and also the beauty of networks, also through art. And if you visit his website, you'll may be surprised to see a kind of 50-50 split between science and art. But I think today he's going to treat us mostly to the science part. Although I might be wrong. Let's see. So without further ado, László, the floor is yours.
Thank you so much, Petra. I'm so glad that you guys restarted this series because I was kind of a fan of the previous one, and now we're back. So it's a pleasure indeed to kind of be here in this long-running one. And let me share my screen so that we can get started with the lecture itself. And here we go. And we should be going. You must be seeing my screen right now, right? Perfect, good. So indeed, as Petra said, I'm a network scientist, and probably to this audience, you don't have to introduce networks, right? But I will nevertheless say that, much of my research in the last few years has been focusing on this particular network, like the protein-protein interaction network within the cell. And the reason I want to actually show because this is kind of embodies many of the networks that we often think about in the broader context of network science, which is kind of like interactions that are transitory. So like people kind of shake hands and then they depart and they don't talk to each other, emails occasionally and so on. And it does not necessarily represent a permanent connection, as this video is kind of illustrating by showing the links disappearing and re-emerging. Of course, in the protein interaction networks, there is some permanence of the way proteins assemble. And this was only a visual trick we used to kind of be able to show the density of the network. But today I want to talk about networks that are not like this. That is, networks that don't have a transitory nature, where the links have actually more permanency. And this is something that we started to call in my lab and in general kind of physical networks. And physical networks are networks where the nodes and particularly the links have a physical manifestation, right? And the most obvious is transportation networks, where you have the roads, right? But also the vasculature, and then kind of the wiring of a computer. But you could also kind of consider the wiring of a computer chip, right? Increasingly metamaterials, an area that I'm very interested in, cytoskeleton and so on. And of course, the gorilla in the room is the brain, right? And because obviously the brain, and I don't have to explain it one to anyone in this particular audience, is a physically wired network where once it shapes its structure, right, the links are there permanently, or at least the structure is there, the neurons are there permanently, and the links strength may change, but nevertheless, the overall layout doesn't. And the question is, why are physical networks special? Are they any different from non-physical networks? And of course, obviously they have a spatial nature, right? So many of the non-physical networks, we don't deeply care about the role of the space, but here it does matter. They're embedded. The nodes and the links are embedded in the 3D space. Very, very important in many cases, particularly if the network is dense, is that there's a volume exclusion, that the nodes and the links cannot overlap. They cannot coexist in the same point in space. So this means that they must avoid each other. They may need to find paths where we don't cross each other. And that may not matter if the network is very sparse, but if it becomes dense, that becomes a very key feature. And the brain is a very dense network. And of course, what I'm going to talk today a lot about is the idea that these physical networks require energy and matter to build. So we believe that there is a cost associated with them, and hence nature, and both technology and nature are relying on optimization principles to do so at a minimal cost. And there are other features also that matter in physical networks, but these are the three that are really uniquely define them. And none of them uniquely, but together actually they go ahead and give us a different picture. And that's why I see different behavior for the systems. And we need kind of a new theory for physical networks. And this is something that I've been actually busy since 2018. It was in 2018 that we published our first paper on the subject, where we just focused on the impact of the spatial exclusion, right? The fact that the links cannot cross each other. And what do I mean by that? The typical 3D or 2D layout software that we use, but let's think about 3D, the force-directed layout does not really care about the fact that the links have physicality. So it's this particular configuration, it's perfectly allowed because we don't assign materiality to the links. However, if you consider that this is material, then you must find a way for the links to avoid each other. And the first work we did actually in 2018 is to kind of do a brute force version of this, which means that you write actually down a Hamiltonian or the energy function, you would call it, that captures all the energies involved in the elasticity of the links, in the node and link interactions, in the node and node interactions, and the link-link interactions. In a way, effectively, you say, I give you a penalty every time you come too close. Either a node comes too close to a link or two links come close to each other. And then the question is how you lay out the network such that this cost would be minimal, both in terms of the length, this is the length, as well as in terms of things don't want to kind of overlap with each other. And if you do that, now you can correct the problem. So like without that particular term, the link-link interaction, you will end up with a situation like that. However, if you turn on the link-link interactions, obviously your software will be much slower, but now you can actually see that the links are slowly kind of avoiding each other. This is a loop that shows you how it's emerging. And then this is now only working at the small scale. Here I'm going to show you a larger network that we laid out, and you can see the computer trying to find the configuration of the network. And you can see kind of not in real time, because this is computer time, how it's laying itself out, right? And finding the three-dimensional structure. You could call this a community finding algorithm. It would be very costly at that, right? But fundamentally, the lowest energy function typically also kind of finds the communities and separates them because that's how you actually get the shortest link length and so on. And then, of course, you end up finding these links. And then one of the fun things we did, since you mentioned art, is that we then end up 3D printing these things, right? And they end up in the museum along with the formula and so on. So that's kind of one way how we reposition the research we do in the lab, occasionally in a different environment. And I think it's really beautiful and striking, the final object that comes out of it. But the question is, if you think about it, what this algorithm does is that it does link length minimization, that is trying to find the smallest link length such that they're not kind of overlapping with each other and the nodes don't collapse on each other, number one. And also it corrects for links not to cross each other, right? Because the shortest path may be for two links to actually cross through, but here it does a correction so not to do that. And fundamentally, this idea or this hypothesis that networks, natural and particularly physical networks, are minimizing the link length actually has a very, very long history. And that's what I'm going to explore next, right? And this history actually goes back to 1926, and I think it was Cecil Murray, I believe she was a British researcher, who has first formulated that in the context of vascular networks. And so where she was trying to understand the branching structure of the vasculature, and she used actually a length and the volume minimization principle, but effectively it was a length minimization what she was using. And then there's a whole literature since 1926 that length minimization is used to look at the neural branch sizes, the length, the morphology of plants, transport networks, supply networks, wiring of the internet, and even the internet trace of Argentine ants and things like that. So these are all networks that are kind of emerge in the physical space. And this simple principle that these networks are trying to build with the minimal length or the minimal cost in this case has been used very, very widely across multiple disciplines. And what's interesting is that behind all of this, there is a very simple theorem, the so-called Steiner theorem or the Steiner graph. And because if you have, for example, 4 nodes, and these are the color, not the blue nodes, that you want to kind of connect them with the shortest path, that is that find a cable that is the shortest, what the Steiner theorem tells us is that that path is not directly connecting the nodes, but actually creating a path where you have to add 2 virtual nodes, the 2 blue ones, and effectively you create a bifurcation in the path. And that particular path is shorter than any path that would try to directly connect the nodes to each other. And the Steiner graph has been used very, very widely by the community, and it makes a couple of very strict predictions. And these are we call them 3 rules because we're going to go after that, right? And the first one is the possibility and the need for bifurcations, right? So you could actually, when you want to create the shortest path between 4 ends, you will actually create bifurcations in the system. In there is also another prediction that you cannot have higher level of degrees. You can only have bifurcations. You cannot have trifurcations. You cannot have 4 nodes coming together because that would be more costly than doing always by pairwise. So this is one strict rule that bifurcations can emerge, but only bifurcations and no higher order splits. The second one is that whenever there's a split, they're all in the same plane. That's a planarity prediction. And the third is that the angle is always fixed, right? And it's predicted by the theory, and it's a very symmetric angle, right? So these are strict predictions. If you really minimize the length, your wiring should obey that. So the first thing we did is that we started to ask, is this true? And I want to emphasize that this was a journey for us, not about the brain, but about physical networks in general. That is, we collected lots of different data sets on different kind of physical networks, and we wanted to see whether these predictions apply. Why is it that this research has been done from 1926 and only we can do it now? Only now we can do that is because the data collection and visualization has gotten to the point that we can have 3D layouts of these networks that previously was very, very limited, only small segments of the vasculature and so on. But now, as you know, we have full brain connectomes, For simple organisms, we have full vasculature again in the brain, and we have lots of data for corals, tropical trees, and so on. So together, they really form a corpus where you can start asking the question, are there common organizing principles? Because these are all physical networks. Is there anything common between them? Or everybody does whatever evolution wants it to do, right? And there's one possibility there's nothing common in between them. And the first way to test it is length minimization really working here? And the answer, it does not. And I'm going to give you the three rules, and I will show you that all of them are actually violated, right? First, let's talk about the bifurcations. Remember, bifurcations are allowed. That's the first thing. They're predicted to exist, but no higher order splits could be possible. And when you look at all of these networks, we find a giant number of bifurcations. There's no problem. That's the branching process, right, in these systems. But we also find trifurcations, right, where a node goes in and three nodes go out. And then we have five-fold splits and six-fold splits and so on in all of these different type of data. And of course, they become fewer and fewer, but they are present. And one of the things we actually showed that they are not the result of resolution issues, that you are just seeing two bifurcations on top of each other. They're really kind of we have many really trifurcations in the, for example, in the brain and in other systems. So that's the first prediction where we are violating, or the first test. We are violating the Steiner principles because we see these higher order splits. The planarity is also giantly broken, because if these would actually be planar, they should all be kind of peaked here, right on the right-hand side, what we call the Steiner one. And when you look at the distribution of the space angles that you have, then they're all over the place. So there's not at all kind of limited to this narrow angle where they need to be. And also the angle symmetry is broken here. If Steiner tree would be true, all the data should be peaked around here, right? And you see a very, very broad distribution all over the place. So this was kind of the empirical data to tell us that not only the Steiner prediction doesn't work, but these type of systems seem to be very, very different from each other, right? Even though there are some common features, but certainly those features are not captured by the length minimization principle. So then the question is, what do you do next? And one thing, when you start looking particularly at these beautiful brain scans, right? This kind of connectome data that is coming out from the connectome community, you realize that these physical networks are really three-dimensional manifolds that have a little rough surface, but fundamentally it's a smooth surface, right? And these are three-dimensional objects. And then the question you start asking, is it length minimization that matters when you have such a robust and thick features happening in the system? So you would say, what could happen? Volume minimization and surface minimization. And volume minimization typically is treated as just kind of a somewhat trivial extension of the length minimization, right? You just replace them with tubes. And that's kind of the standard treatment of that. That was Murray's treatment back then, right? Cecil Murray's treatment. But the surface minimization is a very complicated one, because these are actually surfaces that, from a physicist perspective, they have surface tension, which kind of smooths them out. And the problem is that you cannot really mathematically easily or create these smooth surfaces that connect a set of nodes, because it's a mathematically very, very, very complicated problem. Because fundamentally, what you're trying to do is to really create a smooth surface that is totally differentiable mathematically, right? That can connect, for example, four nodes. So traditionally in the length minimization, we connect four nodes like that. We want to replace it with the tube system, right? Which is minimal now in the surface and not the total length. But we also want to make sure that it's totally differentiable, right? So it doesn't look like that, but it looks much more like that. There are no breaks on the surface. And the way you typically do that mathematically is that you put lots of patches around the surface. Because, if you have a symmetric case, you may find a closed solution, but when you have an asymmetric case, you have to patch it from many, many surfaces, and you have to connect them continuously. For all practical purposes, that appears to be a problem that is NP complete. But it turns out that only appears to be incomplete. I mean, it's still a very complicated problem, and really the break in our research was the realization by C.N.G. Meng, who was the first author of the paper, and this was really his observation and his discovery, is that there is actually a tool set out there already to minimize surfaces. And oddly, that particular tool set comes from string theory. And why are string theory excited about that? Because, in quantum mechanics, and particularly quantum field theory, we're dealing with Feynman diagrams. That is that if you look at the particle, then the particle looks like this is a trajectory of a particle, and interactions or collisions really correspond to these kind of wavy lines between them. So this is a well-studied, this is everybody who had a physics degree had to deal with this, right? As I did. And what's interesting about string theory is that it replaced these kind of trajectories with a three-dimensional surface, which is called word sheets, and really cuts on this surface correspond to the trajectory of the particles in the space of interest for them. So the strings are really the cuts on the surface that you see actually over there. And in order to achieve that, they had to actually figure out how to minimize the surface, because fundamentally the theory works that you have to find a minimal surface and then you have to do cut through that. And they have, given that string theory is a very highly mathematical subject, they have proven quite a number of theorems on how to achieve that. There's a so-called Nambu theorem, which is kind of the foundation of all of that. And in addition to that, they also developed a set of numerical tools that makes it relatively easy to achieve that. So our key discovery was the equivalence of the two problems, right? That the mathematical framework that string theorists have developed can be actually immediately taken back to our network problem and create minimal surfaces. So what's the problem we are trying to solve? We want to have we have three links, right? Who want to kind of connect, or three nodes who want to connect. We know the shortest path between them would be to have a node in between. And then how do you smoothly connect them? You see, how do you take these tubes and deform them to be a smooth surface? And effectively, you characterize now the surface as a variable, right, in the three dimensions, and you define the cost function. And the cost function that we need to minimize in the physical network problem is formally identical to what is called in string theory to the Nambu-Goto action. And don't grill me on that because that's not my field, but it does work, right? And effectively, this is kind of the geometrical problem we are trying to solve. And at the end, if you have three nodes, it will give you the smallest surface that would connect them, but it would also give you the smallest surface that would connect four different nodes. And I should emphasize that there's a constraint that we had to put in there, and it's also, you can do it in string theory, is that we said that there is a minimal diameter. So the diameter cannot shrink to zero ever, and that's imposed in the system. And that is a key feature of that whole solution. If you don't impose it, then it will always just collapse. Right? And that particularly relevant when it comes to the vasculature, because that's just only a tube, right? And to some degree, the brain neurons are also a tube that lots of things are happening, but there's lots of cost to build the cell wall, right? So now that we have this machinery, what can we do with that? And what did it predict? So let me tell you a couple of predictions, right? So one of the predictions is that, for example, whether you have bifurcations very much depends what's the ratio between the thickness of the line and the distance between them, right? So this is kind of the R is how far things are from each other, and W is the thickness of the line. And what it tells you that if the lines are very thin, then you have mainly bifurcations. But once the line becomes thicker, then the two bifurcations join and they become a trifurcation effectively in the system. And not only that they join, but that becomes the lowest energy state in the system. And this is what we're doing here. We're measuring the total surface. In our case, energy is the total surface. And you see that the total surface actually collapses, or that distance between the two nodes kind of collapses and becomes zero. And for a wide range of parameters of W and R, that is the lowest energy solution, right? Or the lowest surface solution. And once we had that, we are actually able to turn that prediction in things that are testable. So you can actually measure the length distribution between bifurcations, and you can test that across many, many different networks from the brain to the blood vessels. And we see that the Steiner theorem does not fit the data, but actually the manifold actually fits very, very well. And so that was kind of the first prediction that was very much now in agreement with the data across many systems. And the second prediction, which was also fun, is that if you have a very thick and a very thin lines kind of meeting, then the angle really depends on the angle of where the small branch will go out depends very much on the ratio of the thicknesses. And remember, in Steiner theorem, we don't have thicknesses and the angle always has to be the same, right? And in here, however, the angle depends on the ratio. And most important, once the ratio is under a certain threshold, that the difference between them is very large in the diameter, then we have a prediction that it always has to go out perpendicularly. That is, in neuroscience language, it has to spout, right? And when you actually look at the neurons, you see that they're full of these 50 degree or like 90 degrees kind of small branches. And this is telling us really that once the ratio between the thick and the small one is over a threshold, it always has to go perpendicularly out. So we think that probably this was the lowest energy solution, and the brain has learned to take advantage of that to form synapses to the nearby nodes because it's very non-costly to, so that lowest cost is really to send out perpendicularly to that. And that angle is really determined by the surface minimization rather than by the biological need. It just goes perpendicularly out because that's the cheapest solution. And so I should kind of finally say that this is kind of connecting to that we see that cost minimization is a stereotypical principle. And we often think many features to be function driven, but I actually kind of, based on this project, started to think that most likely evolution takes advantage of these energy minimization principles and builds function on top of these low energy solutions like the spouting. And just for fun, I should also mention that these energy minimizations are networks kind of have really funny structures, right? If you twist it, then becomes something like that, right? If you kind of build a network, you can have a very interesting and non-trivial structure, like the one that you see in the Nature cover, right? We ended up 3D printing one of these structures and photograph it, and that's what ended up on the object. So if you care more about this, there is that paper, and I put that link actually on the chat. So that is the string theorist perspective on our work. And so if you have 5 minutes after you actually left this lecture, feel free to look at it because it's really fun the way Sabrina has presented our work. So, and I didn't even knew about that particular side, but now I'm kind of a big fan of it. Now, so this was really, everything that I told you so far was really about the microstructure of physical networks. What happens locally, right? And what are the energy principles that kind of lets you, the branches are following and so on. But let's now step back and ask ourselves, what about the large scale structure, right? And how does really the connectome look like? And what can we learn now that we really have data about the connectome, right? And of course, the fact that the brain is a network did not need much convincing these days. This was kind of Ramon Cajal's major discovery for which he got the Nobel Prize, right? But until recently, we really never had data about the brain, right? The only connectome data that was existing for several decades was the C. elegans from 1986. And those of you who have worked with that, and I know there are many in the audience, you know how depressively dense network it is and how small it is and how difficult it is to actually extract any information about the network structure given the peculiarities of this particular system. But nevertheless, people tried, right? And we tried and other people did. The first analysis of the C. elegans as a network, I think, was the Amaral et al. Paper in 2000, who actually claimed that it's a randomly growing network, meaning that it's a network that grows like the BA model, for those of you who are familiar with that, but it lacks preferential attachment. And so that it becomes a pure exponential, and at least that was the best fit. And of course, this is about the connectome. And in the meantime, many of you are actually in the fMRI space. Lots of data became available on the fMRI space of how these networks look like. And there was a consistent discovery, independently reported by multiple groups and then confirmed by others, that that seems to be scale-free. So, and this was kind of the situation until a few years ago, that these two kind of paradigms were fighting with each other. And the question was, what is really happening in these networks? And I'll come back to that, what this paradigm fight means. And of course, the opportunity to ask these questions has changed dramatically, kind of like a year or two ago, when suddenly, within a short time frame, quite a number of connectomes were published and made available, first made available to the community for study, and eventually published. And now they kind of span from C. elegans to the human. And of course, their completeness varies a lot. We do have a couple of complete connectomes, or some, which is the C. elegans, the Flywire data set. Those are kind of, and to some degree, I think now there is anyway, I think these are the two only complete ones. But non-complete data sets, that is just a certain region of the brain, are available across multiple organisms. And they capture tens of thousands of neurons. So these data sets allow us to kind of go back to the question and say, what kind of animal is really the human or the connectome in general, right? And the question is what to expect. And coming back to this earlier kind of results that some people saw scale-free, others actually saw kind of a random distribution. Let's see what this means, right? And in network science, there are two fighting paradigms that we are all using to kind of think about networks, right? One of them is kind of is the random network paradigm, which says simply nodes are randomly connected to each other. The best manifestation is the Erdős and Rényi work, actually, that from 1960, who introduced and studied that model more like studied. But there are also manifestations, what the model A from the BA model, which is kind of growing, but there's no preferential attachment. In one case, you would, in the case of the random network, we expect a Poisson degree distribution. In the case of the model A, we expect a pure exponential degree distribution. And of course, these networks, the common feature of these random networks is that most nodes have comparable degrees. There are no highly connected nodes or hubs. And of course, comes now the scale-free network paradigm, which is the observation that many real networks out there, from the internet to World Wide Web to protein interaction networks and so on, are actually described by a power law degree distribution. And the mechanism for that power law is coming from growth and preferential attachment, right? And that's kind of my claim to fame in the network science space of trying to understand where these networks come from. Now, this is kind of the basis of how we think about that. And then, of course, real networks are characterized by a non-trivial combination of these two features, right? So no network is purely power law or purely exponential, but we see combinations of those. And we know why those combinations emerge, and we can detect it and we can analyze it and so on. But always we have these two boxes to start from. Now, the question is, what is the brain, right? And so in order to understand, let's measure the degree distribution for the brain, right? And now let's do that for this connectome data. Now, given this audience, I'm not going to go into how we analyze this data. About half a year of this project was to clean all of these data sets across these multiple connectomes and bring them on the same place. And this wasn't simply just a homegrown process. We actually had to reach out to virtually all of the groups who put the data out to understand the idiosyncrasies of how they actually collected and put the data out. So that, because if we didn't do that, which at the beginning we didn't do that, nothing looked like anything else, right? But we managed to kind of synchronize these different features and get data sets that finally could talk to each other in terms of the measurable quantities. And so let's talk about the node degrees. And of course, degree is just simply how many links a neuron has in this particular case. And this is the degree distribution we found for these different organisms. And if you look a network scientist and you look at it, you say, holy something, right? because it looks like nothing else. First of all, it does have a peak distribution, which indicates a Poisson, but we are on a log linear scale. So in a log linear scale, the Poisson doesn't look like that, right? So, and of course, they're all a little different. And some of the differences are rooted in the data kind of peculiarities because most of them are not complete data sets. They are just a segment of that. So how do you make sense of all of this, right? And this is where statistical physics tools come in because we in statistical physics have learned to deal with data coming from multiple different systems that may or may not have a common mechanism of action. And the way we would treat these data sets is to rescale them, right? Is to say, is there a possibility that these data sets are coming from the same underlying distribution with a different set of parameters? And you can do that even without knowing what that distribution is. And this is key. I don't have to make an assumption about the nature of the distribution. I'm just going to rescale my data and see what comes out. And how you rescale the data, you're on a log scale. So you pretty much, you rescale horizontally, which corresponds typically to a horizontal shifting in the, so that everybody is in the same average, right? And which means that you calculate simply the average of each data set. That's your mu k. And then you effectively take off this mu k from the data, divided by the sigma, which is the variance of the data. This you can determine from the data without making any hypothesis about that. And then when you do that, and now you're going to plot not the degree distribution, but the rescales variable. So you see horizontally, I'm plotting the rescale degree, virtually just pk in that rescale degree. And what you realize is that they're kind of overlapping and they look alike in this common plot, right? And this is, again, I had made no assumption whatsoever what this distribution is. But we are in a log linear scale, so this cries out for the possibility that you could fit that, right? And in this case, I'm effectively fitting it with a Gaussian on a log linear scale, which a Gaussian on a log linear scale has a particular meaning, and I'll tell you a little bit about that in the next second. So this is the procedure we're going to use. This was about the degree. Now let's talk about the node strength. The node strength, as you know, is simply the number of synapses that two neurons have to each other, right? Previously, I just said how many distinct neurons you were connected to. Now I'm going to tell you how many synapses you have between them. And again, when you look at the distribution of the node strength, again, you find distributions that look alike, but actually now they are much more spread around, which is good because we have more data in this case when you look at the node strength. And now I'm going to do the same procedure. I'm going to rescale them without any assumption about what kind of distribution it is. I'm going to shift them horizontally, and then here now it's even better the fit, right? To kind of a Gaussian in the log linear space. So then let's just put them together what we got, right? We have these two fundamental features of the network. The data sets look all different, but if you just horizontally shift, and that's all I did, I horizontally shifted and then I rescaled their spread to be the same sigma, right? They all collapse onto each other, and the collapse is particularly good for where we have lots of data, which is the strength, and it's a little noisier for the degree because it's a more discrete data. So what do we do next? Well, first, let's talk about what is the fit. I told you that I gave you a Gaussian fit. A Gaussian fit in a log linear space is nothing but what we call a log normal distribution, right? And so let's talk for a second about log normals because we're going to have to deal with that, right? So log normal distribution has this form. Effectively, it's a Gaussian, but the variable is ln k and not simply k, right? If it would be just k, it would be a pure Gaussian. And here we have ln k. And log normals have been used a lot to fit lots of data sets because have an interesting feature. They can be very peaked. If this sigma variable, the sigma is relatively high compared to the mu, then it's a very peak distribution. It looks like a Gaussian, right? And then if sigma is small, and if sigma is large, then it looks like a decreasing function, and it's often used to fit a fat tail distribution. So every time you see kind of a fat tailish and you don't have a mechanism of action, if you try to fit a log normal, it typically does a good job, right? And the problem is that in the network science perspective, you could get a very good fit, but network science has a peculiar focus on the fact that we don't only fit, we need to predict as well. That is, we need to derive the function that we need to describe. And the problem we have with log normal is that in the network science context, when it comes to degrees, we don't have a theory that would justify a log normal degree distribution. You can fit as much as you want. And as I said, you can actually fit lots of power laws with the log normal and would give you a good fit, but you don't have a mechanism of why that should be log normal. And I'll tell you in a second why we don't have a mechanism, right? So fitting a log normal to PK remains a statistical exercise rather than something that we can actually see why is that happening. And why is that? Because when it comes to random wiring, we have models. We have the Erdős-Sérényi and Model A that predicts us in a network context that that distribution has to exist. In the case of the scale-free network, we have growth and preferential attachment and many variations on that predict your power law as well as deviations from the power law. However, log normals are described by a multiplicative process. And network growth is not multiplicative, but it's a gradual, it's an additive process. You add one node at a time, right? And the process is when you're growing something that you're growing in fractions, like you now add 10% more and then 10% more of what you had, right? So that gives you an exponential process and that becomes a multiplicative process. And as far as we know, nobody has been able to predict or to justify that the networks would follow a multiplicative process. So you can fit as much you want, but you don't have a theory of why that would be a valid fit. And so that's a problem because we just concluded that these networks are log normal, right? And whenever we do all the statistical tests, this is noisy data. So it's never a perfect fit, but across multiple data sets, the log normal gives you the best statistics, right? Of course, just doing a statistical fit doesn't tell you anything, but that should be the right value of statistics. But at least it's kind of consistent that log normal typically does across multiple data sets a better job than the alternatives that exist in this system. So now the question is, why is it log normal, right? And now we're going to close the loop of this lecture because I started out with physical networks and I forgot about them. And now it's time to go back to physical networks, right? Because brains are physical networks, right? So we tend to describe the connectome like that. I have neuron 1 connected to neuron 2 with 23 synapses and 2 to 3. But in reality, when you look at, you see this very beautiful sculpture in the space, right? That touches each other in multiple places. And the physicality is the key to the connectivity, right? Because the neurons are physical objects that can only connect to each other if they're physically close to each other, close enough to form synapses with each other. So, and key feature is that while often in network science we distinguish the nodes and the links, there is no distinction in the case of the neurons, right? Because the branches are serving both nodes and links. And of course, it's tempting to put the body of the neuron as being the node, but it's really meaningless when you look at it, right? Because really the neuron is the whole branching structure and the body just has a particular role, but is not part of the communication pattern, right? So the question is, what does physicality mean, right? And we can now characterize each neuron in terms of degree, strength, length, and then density of synapses, which are kind of capturing some of their physical features. And I want to emphasize that these are going to be things that we're going to be dealing with. I already told you about the degree and the strength, right? Degree, how many links it has to other neurons, how many other neurons connects to strength, how many synapses it has to other neurons. And the length is how long is the trace of the neuron, the total length, and the density of how many what's the density of synapses on that particular one. And so the question is, if it's physical, how does physicality matter? And why would it change anything, or is just the same story that we had before? So let's talk about why does physically matter? Well, it matters because for neurons to connect, they have to reach each other. And the longer a neuron is, the more possibility it has to connect to other synapses. So the length of the neuron matters. And the hypothesis you can easily do, a back of the envelope hypothesis, is the number of synapses a neuron has to other neurons is the density of the synapses times the length, right? Very trivial assumption. And it's true as well, right? When you looked actually of how many synapses neurons have in terms of their length, and these are the different organisms, and each circle corresponds to a different neuron, you see that there is this asymptotic behavior that the longer they are linearly, the more synapses they have. It's a no-brainer once you realize that this is a physical network. And the question is, why does physicality matter? Because in order to understand now this number of synapses and the degree, you have two covariates, right? You need to know the length and you need to know the density of the synapses. So now we have to ask ourselves, what's their distribution, right? How are they distributed? And so this is the distribution we can measure in the data, right? Whenever it's possible here, we don't have data, but for these we have, and they look like the previous data set. So not surprisingly, when you rescale them, they again follow as good as possible in log normals, right? And so now let me give you a little primer on log normals because we're going to use that. So log normal has this particular functional form, and each log normal has two variables. One is the average and the other one is the sigma, just like a Gaussian, except that these are now defined in the log space. And what's interesting about the log normals is that if you have two variables that each follow log normal, their product would also be log normal. And not only their product will be log normal, but if you know the parameters of the x and y, then you can precisely predict what would be the parameters of the products, right? And there's a simple formula to tell you that. It's and also if you have, let's say, a function like that, and x follows a log normal, then the y distribution will also follow a log normal with parameters that can be analytically predicted, right? So there's no room for playing of if these things are connected, of what the relationship between them. And so, but the neurons, I just told you that the length and the rho are log normals, and we're predicting that the synapses should be rho times L. So if these two follow log normal, then the sinus distribution should also follow log normal. And not only that, but I can now analytically calculate what should be the average and the sigma for the sinus distribution, only knowing the length and the rho of the neurons, right? So I'm making a prediction about one measurable variables from two other measurable variables. And these are only characterizing the physicality system, and these are the connectivity. And those predictions work, right? So this is the measured and this is the predicted for the different organisms. And you see that they are kind of fitting very, very well, and also for the sigmas. So both of them are actually, the data follows that. So this kind of shows it's called a closure equation, right? Because if these follow, then the sum and the product should also follow, and you see that that's indeed the case. So we can make predictions about the neurons. And then here I'm showing you the synapse distribution for three different organisms. And I want to emphasize the line is not a fit. The line is a prediction, right? The line is predicted just based on the neuron lengths, and the density of the synapses. By the way, the density of the synapses is very constant across organisms. So there's very little variations. There's even a paper recently by one of my co-authors who was a biologist who actually kind of showed that. And we also saw it in our data. We just didn't go after that, but it's a very interesting observation by itself. So it's really only the length that drives these things. Good. So let's go back to the degree distribution, because this was about the strength. And we observe in the data that there's actually a power relationship between the degree and the strength. And eventually we were able to also derive it analytically. So that's kind of in the paper. So I'm not going to go into that. But fundamentally, if this relationship is a power law, then we can predict what should be for the degrees, the mu and the sigma. That is, I can analytically predict the degree distribution. And again, just to show very briefly that these predictions work. That is what we predict from the neuron length actually predicts very well the degrees. And sorry, and I jumped too fast. And I want to emphasize again, this is the degree distribution, and the lines are not fit, but predictions. And it shows how, as good as it gets in this particular story. So now let's go back because time is passing, and let's kind of address the main issue, right? Because I have now convinced, hopefully you, and certainly convinced myself that the empirical data indicates that the brain is a log normal network, and we are still in the trouble because we don't have a theory. And to the contrary, no, we don't have any mechanism by which log normality or multiplicative processes could emerge, not in the brain, but in any network context. And that's the key, right? In any network context, no one has been able to really credibly show that there is a generic mechanism that could actually provide multiplicative processes. So what do we do in this case? This is a dead end. Well, not. So what I'm going to show you now, obviously, is what we think that the mechanism here is. And that again comes back to the physicality of these networks, right? So each neuron is a physical object, and most precisely, it's a tree. And now I'm going to exploit the tree nature of this, right? Each neuron is an incomplete tree. And what you see on the right-hand side is exactly the same on the left-hand side, except I'm now showing you only the tree structure, the branching structure of that system. And you see it's an incomplete tree because some branches start and they dead end early, and others go as far as they can get, right? So what does that mean? It means that we can actually describe it as an incomplete tree. And actually, Watson and Galton in 1874 have ended up describing that. Yet another British result, right? What was the motivation? They wanted to understand why do some certain noble families die out and others survive. And so they kind of looked at this as it's a survival process. You have a kid, and she or she may not have a kid, and then it dead ends, or it will continue the branch. And so each node has a survival probability, and ultimately that the total length of this branch, or the length of your family, right? The size of your family tree is really the product of each of these individual probabilities. And we have a multiplicative process. So a tree is really driven by a multiplicative process. Now, what do we do with this multiplicative process? First, we have something, right? So let's look at the model. So if the survival probability is less than 1/2, then the branches will die out very fast. However, if they're larger than 1/2, they will still die out they can still die out with a certain probability. And you can analytically prove, and we did that actually, that in that case, the length distribution of the branches will follow a log normal distribution. And not only that we actually proved analytically that has to be log normal, but we can now connect the parameters of the log normal to the probabilities that are characterizing the branching process in the neurons. And as I'll show you that next, those probabilities are measurable. How are they measurable? The survival probability is just simply how many branches are from one layer to the next one, right? How many branches continued and how many branches died out. And that's so simply, if you go layer by layer, you can measure in each layer the survival probability. And this is how the survival probability starts. It starts out very high, then it drops at layer 2, right? And then it kind of stays strictly in the superlinear regime, larger than 0.5, but approaching 0.5. And that is key because that's why they end, right? As you get close to 0.5, you will have to end. And so now this is no longer a hypothesis. The survival probabilities can be measured explicitly for every single level of every single neuron in the connectome data. So here I'm showing you actually this distribution, the strength distribution and the neural length distribution for not only for the whole neuron, but different regions of the brain. First showing that they all look normal, so it's independent of the brain region. But I'm showing you for each classes of neurons, what are the survival probabilities, sorry. And they do depend on the class, and that dependence actually determines whether the neurons are big or small in that class, or they have lots of branches or not. But fundamentally, we can analytically calculate now from the survival probabilities, from the morphology of the neuron, effectively all the parameters of the log normal. So this kind of closes the loop because we were now able to simply get parameters from the branching process and predict from there everything going back, the strength distribution, the degree distribution, and the length distribution of the neurons. So we can go back and predict the large scale structure of the brain. And so now that we think we closed the loop, then the question is, why do we care? Right? That is, why do we care this is log normal? Right? Is it any different from a random or from a scale-free? We know the random and the scale-free are giantly different, but what about log normals? And of course, log normals are exactly in between, right? In some ways. Sometimes you can look at fat tail, depending on the parameters, sometimes are there. And you can calculate all of its parameters, just like average degree, second moment, sigma, maximum degree, and kappa ratio, and so on. You can analytically calculate all of that. And then let me show you why it matters. First, we need to say, why does the scale-free feature matters? Because scale-free networks have this weird property that their second moment is infinite, right? Which means that there's a giant variability in the system, which is due to the hops. The hops are so highly connected in the system, and the second moment is infinite. And that the fact that the second moment can become very, very large, even if it's not infinite, it's very, very large, has lots of consequences. For example, when it comes to the robustness of the networks to node to like link removal or node removal, when it comes to spreading processes, when it comes to synchronization, in all of these areas, the second moment matters, and the second moment becomes very large. Then either the critical threshold becomes converging to one, or the epidemic threshold becomes zero, or the synchronization threshold becomes zero. So fundamentally, this high variability that we have in scale-free networks fundamentally changes the properties of the system, right? And the dynamics of the system. And for the brain, for example, synchronizability and network robustness are very important, and those are affected by the hubs. Well, what happens if we don't have scale-free networks, right? Well, let's look at what the second moment or the first moment looks in a scale-free network in systems that are comparable to the brain, right? So here I'm showing you the brain size. Humans are somewhere here. The C. elegans is here. And you can see if you have a random network, this is flat. There is not much variability. In a scale-free network, this grows like a power law, right? So it becomes huge, but in a log normal also becomes fat, which is telling us that in the log normal network, the variability is finite. So the robustness and the synchronizability of the connectome does not depend on the size of the brain. So this is very interesting, even for myself. We have never calculated these features because we had no reason to look at log normal networks because those animals don't exist in networks, right? But now that we did the calculation, we realized they have these very unexpected features. So they have hubs, they have highly connected nodes, and I'm showing you here that they have, right? So when you look at the maximum degree, the log normal has a much larger maximum degree than a random network, but not as large as a scale-free. And this also makes sense because, for example, we know that some of the most connected cells in the brain is like the Purkinje cells with 200,000 synapses and about 7,600 degree. But if the network would be scale-free, you should expect a 10 million degree node in the human brain, right? And we don't see that. But if you actually say, let's look at the log normal, it predicts you about 10,000, which is very, very comparable. So the log normal can account for the fact that you have highly connected nodes, much more connected than in a random network, but not as big as actually you have in the scale free. And it's very much in line with what we know so far about how, for example, the human brain looks like. So I'm going to end here. I think my time is slowly up anyway. And what I was trying to tell you really on one end in the brain spaces, we're finding that the brain is different, right? And of course, most of you knew that, right, instinctively, but we didn't know how they're different. But fundamentally, what we are finding is that if we treat the connectome as a physical network, and we kind of go beyond that simple graph theoretical decision that we have nodes connected by links, but we consider the three-dimensionality of these networks, then somehow the data makes sense, right? And we can actually now predict the type of distributions that we see in the empirical data. And from a network science perspective, I'm very excited about that because this is really a new class of networks. As I said, people have had a long history of trying to fit log normals to data without a justification, because statistically you can fit not anything you want, but sometimes you get a better fit if you fit log normal. But the reason we never accepted those fits in the network science space, but because we did not have a theoretical justification. Why would they emerge? Because networks are not multiplicative systems. Neither is the brain a multiplicative system, but the neurons are, right? And that's the key. The branching process of the neurons defines a multiplicative system. That process defines the length of the neurons. The length of the neurons, because this is a physical network, drives the connectivity, the degree, and the number of synapses. And that's how we get the full story. So hopefully, now you saw the story going from the microscopic branching processes, from surface minimization, all the way to how do we think about the large-scale structure of the system. And here are the co-authors of these two key papers, and the two papers are here. One of them just came out 2 weeks ago in Nature. The other one still kind of working its way through the referring process. And of course, I want to acknowledge all my collaborators who made this possible. And thank you very much for your attention.
Amazing. Thank you so much. I'm trying to find the clapping emoji. There we are.